And we are back with the babies. You can tell I like them a lot. They're so cute. People are so diverse with all the cool traits that they have. Different colored eyes, and like you got different hair texture. You got the wavy and curly, and got the straight hair babies, and all different kind of heights and structures to them. So how does that happen? I'll move this up. Here's some cool traits that are genetic. Even got the straight teeth and the gap teeth. That's a genetic trait. You can take a look at your toes. If your second toe's longer than the big one, that is a genetic thing you got from your parents. Got any burrito tongue, like tongue rollers, that's a genetic thing. So lots of cool stuff there. How do those things get coded in the cell is what we need to focus on. What is this coding that happens? When does it happen? So first we're going to do the when. Cells operate on a cycle, so they're always in some point in this big cycle, this circle that's going on. We've already talked about the mitosis, the cell division, making one cell into two, and you can tell how the whole rest of this circle is much bigger than this little piece of the pie here. So all of this is called interphase. That's that preparation we already talked about. The preparation for the game, most athletes would tell you that is probably the most important part. So the preparation for a cell divide is the same way. So all of this, G1, S, G2, this big piece of the pie, all of that, those are three stages that happen during interphase. So a cell is doing a lot of stuff to prepare to divide. And that is where the coding comes in. Coding is very important. Let's look at these three first. G1 is the first part of interphase. That's where you're going to make copies of some important organelles that we need. S is our focus for this lesson, really. S is where you are going to make copies of the DNA. If you see here, you see this one little squiggle that's red and one that's blue. From here to here, the squiggle turned into an X. You have a red X and a blue X. So they made copies of themselves. That happened during S phase. Also, the coding for your traits. The proteins are going to be made right here in S. G2 is another growth phase, and they're going to check for errors and things before it lets the cell divide. So interphase is where the magic happens, the most important part. So that is when stuff gets coded. Just a overall picture here of that. You can tell interphase has just a single line chromosomes and that single set. Like you have this single line, that's a chromosome, and you have one pair of centrioles to help the cell divide. And before it got to prophase, it made copies. So now DNA, these chromosomes have copied, and the organelles have copied. So that's what happens during interphase. Now we're going to zoom in to those chromosomes and see what is so cool about them. How does this coding happen? So if we were to just pull one of those chromosomes out of the nucleus, make it big enough to actually see and unravel that thing, you would see it is made of DNA. DNA is the key to everything. It's the key to life. Everything in a cell operates based on its instructions. It is the boss or the brain of a cell. That's where the coding comes in. I'll scroll this up. DNA, just like if you did computer coding, if you're one of those that know a lot about coding for computer hacking, it has its own language. DNA has its own language too. Let's do a little DNA structure 101 first so we can figure out this language. Turn that. DNA stands for deoxyribonucleic acid. So in the name, it says it is a nucleic acid. So it is built of monomers or building blocks that are called nucleotides. So these three parts you need to know are three parts that make up a nucleotide. Phosphates are always circular, so that's our phosphate. It's bonded to this five-sided house looking thing. Anytime you see a five-sided shape like a house, it's going to be a sugar. This one is called deoxyribose. So the ose ending tells you it's a sugar. And then that's bonded to a base. Now this is kind of a generic shape here because you're about to learn that there are 4 different bases and they all have different shapes. But this is generically, if I were to say what are the 3 parts of a nucleotide, you need to be able to say phosphate and the sugar and the base. To me, it kind of looks like the Up House. If you watched that that Disney movie, it has the little balloon attached to the house. And then we have the base here. Here is the structure that hopefully you recognize as DNA. They call this a double helix sort of structure because it's all twisted and wound up at each other. So if we were to like take this piece here and just flatten it out and try to make it to where we could actually see what it was and zoom in a little more, this is what it would look like here. Here's your Up House. You got the phosphate and the sugar, kind of a repeating thing on the left side, phosphate, sugar, going all the way down the left side, and each one of those is bonded to a base. There are 4 bases in DNA, and you need to know what they are. They have long names like adenine, thymine, cytosine, guanine, but a lot of times in these shapes that you see, you just see the first letter for them. Because here you see a T. Maybe you don't see it. I need to... Yeah. Here's a T for the thymine. It has this pointy shape. Then you see A, it has like the opposite to that. That is because A and T are complementary bases. They have a particular shape so that they can be bonded to each other. So look at these, kind of like puzzle pieces. T will only bond with and fit with A. And even if they're switched, you can see A still is only going to fit with T. Here's a third base, C for cytosine. It has a very different kind of little block shape that's in there. It is going to bond with this. It has the block sticking out, and that is a G. So you can see it maybe better down here. This thing sticking out is going to fit right in there. So it's just like puzzle pieces. A and T are complementary bases, and C and G are complementary bases. Hopefully you could see that A would never fit. You would never try to bond it with one of these. They would never fit. So certain bases are complementary to each other. The way I remember that is A goes with T, apples in the tree, and then C goes with G, cars in the garage. So apples in the tree, cars in the garage, complementary bases. I don't know if that whole word will fit in here. Nope, complementary bases. You got A with T, and you got C with G. And y'all can't see that. So you need to know which base goes with what, because that is going to tell us how to replicate our DNA. Here's a cool picture to show you what is happening, because not only do you need to know how to decode the language, but you need to be able to answer questions that are kind of asking what is happening. So if you take this original DNA and an enzyme would come right here and cause it to unzip, like if you had a zipper and you unzip it, it opens up. One side of this DNA would be on this side, and the other side would be down here. So you open it up and you have half of it up here and half of it down there. So how's it going to make a copy of itself? Since we just saw that A always goes with T and C always goes with G, if I open this up and this is a T down here, the cell has other nucleotides available to bring an A in and match it with that one. Just like it would have another T available to come up and match with the A that's up here. So when it's finished, this is kind of what it looks like. And got my thing turned. This is the initial DNA strand. See G going with C and T going with A. It unzipped. So then you have these red ones that are new. A new C came in and matched with that G. Then you also had a new G come in and match with that C. so that when you're finished, you have these 2 strands, but they're going to be exactly like the first one because of the complementary base rule. G will only go with C and A will always go with T with DNA, no matter what. So back to this picture here, this is what we're talking about. It had this little red chromosome. It replicates or copies itself and ends up looking like that. So A going with T, C going with G. This is what it's doing, making copies of itself. What we're going to do now is that breaking the code sheet that I said you should download before watching this. So if you don't have it to where you can write on it yet, you can pause this video. You can either pull it up on Cami and have two frames up on your Chromebook at the same time so you can see me and write on it, or you can print it out and write with me. But this section is going to be kind of like math, so you'll want to have that. We're going to do just this top part. I can't turn that. DNA replication. And hopefully you're going to think this is just the easiest thing you've ever done. We're going to start just by writing the rules that we just went over. And I'll probably just write that here. A goes with T. And C goes with G. And that's vice versa. So G is going to go with C. I'll put it up here. T is going to go with A. So if that's the rule, we're just going to follow it. This is DNA strand number 1. And what you're seeing is just, it's giving you half of that strand, like the top half. And you're just going to match bases for the bottom half. So T goes with A. A goes with T. C goes with G. I'm going to just keep following the rule. C goes with G. G goes with C. G goes with C. A goes with T. T goes with A. G goes with C. G, G, T. C, T, A. G, T, T. G, A, G. Alright, so what I would do is pause the video and take a minute to do DNA strand number 2 and number 3. Just try that on your own and then come back and compare it to the answers that I show you on here. If you have any questions on how to do this, you know what to do. You can email me or send me a remind text, whatever works for you. Because you have to know how to do this decoding before you can do the little spring bunny assignment that comes after this. Okay, so I'm just going to go ahead with this. Hopefully you've already attempted it. I'm going to do this so you can see the key and see if there's any issues. I'm just matching bases. There's DNA number 2, number 3, okay, so we just replicated DNA, breaking the code, and that is just section 1.